A single finger bone. That's all it was. A fragment of a pinky finger, pulled from the sediment of a Siberian cave in 2008. It was so small, so unremarkable in appearance, that it could have been catalogued and shelved without a second thought. But when scientists extracted DNA from that fragment and ran the analysis, the result didn't match anything in the known human family tree. Not Neanderthal, not modern human. Something else entirely. Something that, until that moment, no one had any reason to believe existed. That cave is called Denisova, tucked into the Altai Mountains of southern Siberia, near the borders of Russia, China, Mongolia, and Kazakhstan. Archaeologists had been working there for decades. It was already known as a site with a long human presence, layers of occupation stretching back hundreds of thousands of years. The assumption going into the 2008 excavation season was straightforward. They would find more evidence of Neanderthals or early modern humans, the two groups known to have moved through that part of Asia. What they found instead forced a fundamental rewrite of the human story. To understand why that finger bone was so disruptive, you have to understand what the scientific consensus looked like before it. The accepted framework held that two main groups of archaic humans had populated Eurasia during the Middle to Late Pleistocene, Neanderthals in the West, and modern Homo sapiens spreading out of Africa. There were hints of other populations in the fossil record, unusual skulls, ambiguous jaw fragments, but without genetic data, those anomalies were difficult to classify. The working assumption was that the human family tree, while complex, had a manageable number of branches. The Denisova finger bone didn't just add a branch, it revealed that an entirely separate lineage had been living, moving, and interbreeding across Asia for potentially hundreds of thousands of years, and that science had simply missed it. The first attempt to make sense of the bone came through mitochondrial DNA analysis. The results were striking enough on their own. The mitochondrial sequence was roughly twice as divergent from modern humans as Neanderthal DNA typically is. That suggested a very deep split in the human lineage, far deeper than expected for a population living in the same general region as Neanderthals. Researchers at the Max Planck Institute for Evolutionary Anthropology published their initial findings in 2010. The conclusion was careful but clear. This was a previously unknown hominin. They named it after the cave, Denisovan. But mitochondrial DNA only tells part of the story. It passes through the maternal line and can be misleading about overall population relationships. The turning point came when the team succeeded in sequencing the nuclear genome from the finger bone, a far more complete picture of the individual's biology and ancestry. That analysis confirmed the Denisovans as a distinct population, a sister group to the Neanderthals, both having diverged from the lineage leading to modern humans somewhere around 400,000 to 600,000 years ago. And then came the finding that changed everything about how we understand human prehistory. Traces of Denisovan DNA are present in living people today. Not in Europeans, not in Africans, but in populations across East Asia, Southeast Asia, and most strikingly, in indigenous peoples of Papua New Guinea and Australia, some of whom carry roughly 4 to 6% Denisovan ancestry in their genomes. What that percentage means, mechanically, is that at some point in the past, likely multiple points, modern humans moving through Asia encountered Denisovan populations and had children with them. Those children survived, reproduced, and their descendants eventually became the ancestors of people living today across a vast stretch of the Pacific. The Denisovans weren't a dead end. They contributed to the human gene pool in ways that are still biologically present in millions of people. One of the clearest examples of this is a gene variant associated with high-altitude adaptation found in Tibetan populations. Research has traced this variant in the EPAS1 gene, which helps regulate the body's response to low oxygen, to Denisovan ancestry. It appears that modern humans acquired this advantageous adaptation not through their own mutation, but by inheriting it from a population that had already been living at altitude for a very long time. The cost of this discovery, in scientific terms, is the collapse of a simpler model. For decades, paleoanthropology operated with a relatively clean narrative. Modern humans left Africa, encountered Neanderthals, and eventually replaced them. The Denisovan evidence forces a much messier picture. Multiple archaic populations were present across Asia simultaneously. They were not isolated from each other or from incoming modern humans. They mixed, they exchanged genes, and the genetic legacy of those encounters is distributed unevenly across the modern human population in ways that researchers are still working to fully map. There are significant uncertainties that remain. The physical appearance of Denisovans is still largely unknown. Only a handful of confirmed physical remains have been recovered from Denisova Cave itself: the finger bone, a few teeth, and some small fragments. The teeth are notably large, larger than those of Neanderthals or modern humans, which suggests a robust build, but that's a limited inference from limited material. More recently, researchers have proposed that several fossils found at other sites in China may represent Denisovan individuals or closely related populations. The Harbin cranium, a massive, well-preserved skull found in northeastern China, has been analyzed using mitochondrial DNA and protein evidence, and the results suggest it may represent a Denisovan. A study published in September 2025 proposed that remains from 6 additional Chinese sites, including material associated with the roughly 1 million-year-old Yunxian man, may be related to the same lineage. Other researchers have placed fossils like the Xiahe mandible, found on the Tibetan plateau, and the Penghu 1 jaw from Taiwan within a proposed species designation. These attributions remain under active scientific debate, and no formal species name for Denisovans has yet been established. What is not in dispute is the geographic scale of the Denisovan presence. The genetic evidence places them across a range stretching from Siberia to Southeast Asia, and possibly further. According to the available evidence, they ranged across Asia during the Middle to Late Pleistocene, approximately 200,000 to 32,000 years ago. That's a population persisting for the better part of 200,000 years across an enormous territory, leaving almost no physical trace that science recognized until a fragment of a finger was analyzed in a laboratory. Today, the Denisovans occupy a central and still expanding place in human origins research. Every new ancient genome sequenced from Asia has the potential to add detail to their story. Every unusual fossil from the region is now evaluated against the possibility of Denisovan affiliation. The field has moved from a two-population model of archaic Eurasian humans to something far more complex: a web of populations, migrations, and interbreeding events that played out over hundreds of thousands of years. The lesson the Denisovan finger bone delivers is not simply that there was another kind of human. It's that the absence of evidence is not evidence of absence, and that the tools we use to ask questions determine the answers we're capable of finding. Paleoanthropologists had been excavating Denisova Cave for years before the genetic analysis was possible. The bone was there. The population it represented had been there, across Asia, for millennia. What changed in 2010 wasn't the past. What changed was our ability to read it. And the question that finding opens, how many other populations, how many other chapters of the human story are still waiting in the sediment for the right method to come along, remains entirely unanswered. Visual reconstructions are illustrative. Facts and interpretations are based on the sources listed in the description.